Yep.
Okay, welcome back everybody to the Astrochemistry webinar. Today we are going to be hearing a talk from Dr. Michel Nuevo from NASA Ames on the formation of building blocks of life in astrophysical environments. So just to introduce ourselves to those of you who are new here, I'm Katie Slavachinska, my co-host is Brian Ferrari, and we are chair and vice chair at the University of Central Florida American Vacuum Society. So that's the organization through which we are hosting this webinar. The format is a 45-minute talk by our speaker, followed by a 15-minute Q&A session. You can write your questions in the chat throughout the talk or after the talk. And after Dr. Nuevo is finished presenting, he will go through the chat and answer your questions. So today's speaker is Dr. Michel Nuevo from the Astrochemistry and Astrophysics Laboratory at NASA Ames. And he's going to be giving a talk on, like I said, the formation of the building blocks of life in astrophysical environments. So Dr. Michel Nuevo grew up in France, where he studied physicochemistry and astrophysics at the universities of Orsay and Pierre at Marie Curie in Paris. He obtained his master's degrees in both molecular physicochemistry and instrumentation in astrophysics in 2001. And he obtained his PhD degree from the University of Orsay in 2005, doing his research at the Institut Astrophysique Spatiale in Dr. Louis de Hondecourt's laboratory. After his PhD, he was a postdoc at the Graduate Institute of Astronomy and the Department of Physics of the National Central University in Taiwan for 2 years, where he worked with Drs. Wing Huan Ip and Tai Son Yi. Finally, he arrived at NASA Ames in Dr. Scott Sanford's Astrochemistry laboratory in 2007, where he started as an NPP postdoc fellow. He has been working in the astrochemistry laboratory team since then, now as a Baer Institute research scientist since 2013. So I'm going to go ahead and stop sharing, and Dr. Nuevo, you can take it away.
Okay, let's start to share the screen. This one. All right, can everybody see this? All right.
I don't see the, oh, there we go. I can see
Okay.
it.
All right. So I'm going to do this. Okay. So, so thank you for the introduction, and thank you for inviting me to talk in this series of webinars that have been very popular since it started a few weeks. I would like to talk to you about what we do in our lab astrochemistry group at NASA Ames. And so as an introduction, what we're trying to do in our lab is basically trying to simulate in the lab experimentally the chemistry that is taking place in very cool astrophysical environments, such as the ones you can see on those pictures, for example, like dense clouds, but also star forming region, and also cold parts of protostellar disks. And for this, in the lab, we need to simulate conditions such as very low densities of pressure, very low temperatures, down to 10 to 80 K, and also very high radiation levels, in particular from energetic photons or particles such as electrons. And also we simulate like large temperature fluctuations, warming up the samples to simulate the heating process when the star forms. And how do we know what those astrophysical environments are made of? By studying them through astronomical observation in the infrared range. So you can do that from ground-based observatory, but also some instruments on SOFIA, which is a stratospheric plane with a telescope, or from space telescopes, you can determine what is the composition of those cold environments. And for example, here, you can see that those environments are usually made of cold dust grains, usually made of silicate. You can see here on this infrared spectrum here, but it can also be like carbon-based grains. And as you can also see, that those environments are made of a lot of small volatile molecules that are actually condensed on the surface of those cold grains. And you can see here the list of molecules here on this spectrum. You have water, methanol, ammonia, carbon dioxide, carbon monoxide, and so on, methane, for example. And I'm going to talk about this XCN and OCN minus band a little bit later. And so if you look at several different objects, cold objects, you can see that first that water is the main ice component, but also you can see that there's a lot of molecules that are based on carbon, and this is very important for organic chemistry and for the rest of my talk, because from those molecules we can build complexity, molecular complexity. And I mentioned about this compound here, OCN minus, that was detected in some of the sources, and it's very important because this molecule is basically made from chemical reactions from some of the other ones, and that's kind of a proof that chemistry is actually happening in those cold environments, although the temperatures are very low and densities are very low, but chemistry does happen, and molecules that OCN minus are the proof of that. So the way we see how those processes happen in those cold environments is that you can see here the silicate grain, and you have all those like volatile molecules, but also like even larger molecules that will condense on the surface of those grains, and they will be processed by energetic radiation. So here this is UV, but it could be also energetic electrons or protons. And because of this this energy that those radiation brings, then you're going to have like a lot of chemical reactions, breaking bonds of the molecules and creating reactive species such as like ions. And you're going to build up like a more and more complex material, and usually that more and more complex organic molecules. And why is that important is because those grains with those organic materials are going to be part of the material that is going to be incorporated into objects such as comets and asteroids, or stay in the form of cosmic dust when stars and planets form. And so those grains are going to be participating somehow in the seeding of planets with organic material. And so most of the material that rains on planets, including Earth, every day, even now, comes in the form of meteorites. So you can see here on the left that usually come from asteroids, or what we call interplanetary dust particles, or IDPs, that may come from comets. And so we live actually in a very interesting time, as Scott Sanford told in his talk 2 days ago, is that right now we have able to analyze those materials in the lab after they return to Earth. And so we're going to be able to analyze them for different reasons, but we are more interested, for example, in the organic content. This is something that will happen in the coming years. So to simulate those environments in the lab, we need 2 things. We need sophisticated equipment, like you can see here, and very serious scientists. And so this is a close-up of the chamber and the lamp. So everything, all the chemistry happens inside of this small vacuum chamber here. Inside there's a cold finger with a substrate, and they are cooled down with a cryocooler down to temperature of 10 K. And of course, the chamber is pumped with a turbomolecular pump that is down here. What we do is that we mix gases ahead of time on a different equipment, and we're going to connect those gas mixtures, and they're going to be deposited on the cold substrate through this tube here, the gas deposition line here, and there will be at the same time we're depositing, we're going to irradiate those samples, for example, with the hydrogen UV lamp here that emits photons mostly in the Lyman alpha range. And we are going to process those ices at the same time we deposit them. So here an example of a very simple ice mixture of methanol and ammonia. And the blue spectrum is before irradiation, and you can see that after we irradiate this ice, you have new compounds form, new bands showing up. And if you analyze those bands, you can see that we form a lot of different kinds of chemical functions like alcohol, amines, carboxylic acids, also like aliphatic carbon chains. And you can see also that we make small, very reactive molecules that usually are not stable enough and will react very fast. But since those experiments are made at low temperature and those compounds are trapped in the ice, you can still see them. And also you can form a lot of small organic molecules that will be the building blocks to make even more complex molecules. After we deposit and irradiate those ices at a low temperature, so we usually stop the deposition and irradiation, and we start warming up the chamber to simulate like heating from a nearby star, as I mentioned earlier. And some of the compounds that are formed during those irradiation don't show up until pretty late because they are trapped in the ice. And as you can see here on this plot, you can see a peak here around 250 K when we warm up. And this compound was identified as carbamic acid, which is literally the smallest amino acid because it contains one amino group and one carboxylic acid group. And we did this experiment also with carbon-13 to confirm that the mass was shifted, and it did. So this is the kind of compound that actually can form in those experiments, but that will evaporate before we get to room temperature. And so we won't see it at room temperature, but we know its form during those experiments. So when you reach room temperature and you look at what's left on the substrate, this is what you get here. So it's a very brownish, yellowish kind of material. It looks a bit like goo. And actually one of the pioneers of lab astrochemistry, Meyer Greenberg, used to call this yellow stuff. And he kind of described it pretty accurately. And if you desorb this material with like an infrared laser and look at what's coming out and try to record the mass of the molecules coming out. So here on the right, you can see that from 50 to about 350 molecular masses, you have a peak for almost every single mass here. So these residues are made of a mixture of a lot of different molecules. I don't know, hundreds or thousands of them. And mostly we suspect that all those molecules are organic. So this is another kind of residue that was made for a very specific ice composition to mimic Pluto's surface a few years ago by Dr. Chris Matteries, who is going to talk next week, but not about this. And as you can see, this material is even like darker, like kind of red. And it's very interesting because the color looks pretty much like what we see on the surface of Pluto. So if you analyze those organic residues with infrared spectroscopy, you can see that they are made of a lot of different chemical functions, alcohol, amines, carboxylic acids, carbon chains, nitriles, esters, ketones, you name it. But the only compound that was formally identified in those residues is this one. It's called HMT, or hexamethylene tetramine. And so this is the structure of this compound. I'm going to talk about it very quickly. This compound is pretty interesting because it was first identified in a residue by Max Bernstein in 1995, and by other people too, like here in this plot. So this is the infrared spectrum of a residue made from a mixture that is written there. And what is very interesting that this HMT is always showing up in residues when the starting ice is contained ammonia and methanol. And all those infrared bands that are highlighted with those red squares are actually bands that are attributed to HMT. So we have a lot of it, and sometimes it can, the amount of HMT in a residue can be like up to 70% of the residue is made of this molecule. And why is it so interesting is because sometimes HMT is referred to as organic compounds in a box. And the reason for that is that if you degrade this molecule, either by heating it or like irradiating this molecule with UV, or by hydrolyzing this molecule, you can form a lot of different compounds, organic compounds. In particular here, you can form amino acids, which is very interesting. Most recently, we also found out that some residues contain also derivatives of this HMT. For example, this HMT methanol. I'm not going to go into details. You can refer to this recent paper from Chris Matarese. And we found out that this molecule is formed also in ices that contain methanol and ammonia. And we did a series of experiments with different isotopic labeled ices to determine its structure. And with the help of Dr. Partha Bera and Tim Lee at NASA Ames, who did a DFT calculation of different variants of this HMT molecules with different groups attached to this molecule, we found out that those two infrared bands here are common to all those molecules, and maybe this could be a way to search for this family of molecules in space, thanks to those two bands that are common to all of them. And why is that interesting is because since HMT is known to degrade into a lot of very interesting organic compounds, the fact that there are different also variants of HMT may be also a source of a lot of other also different organic molecules. So what do we know else about those organic residues? So a few years ago, we did some X-ray absorption spectroscopy at the ALS in Berkeley to analyze those residues. And what we found is that it's not a surprise that those residues are made of a lot of different chemical functions, and those confirm the infrared results. But also this analysis also showed that we could determine the elemental composition of those residues, in particular the amount of nitrogen and oxygen compared with the carbon. And by measuring those values, we realized that our residues are usually 2 to 3 times richer in nitrogen and oxygen than stardust particles. So those are properties of the residues as a whole. But about, I would say, 20 years ago, the way to study those residues changed thanks to astrobiology. And instead of trying to figure out what each of those peaks that we saw in the mass spectrum earlier, try to determine what each peak is and what kind of molecule it is, the approach was different in the sense that we started to wonder if those residues contain a certain family of molecules, no matter how abundant they are. The question was, do we have amino acids, for example? And why amino acids? Because they are of astrobiological interest. And so amino acids are the building blocks of proteins, and life on Earth uses 20 of them mostly. And we know that a lot of them, more than 70, have been identified in meteorites. And as I'm going to show you, we also have those amino acids in lab residues. And so to analyze those residues to search for amino acids, we need to use a very specific protocol. And usually we use what we call chromatography, so either in the liquid or gas phase. And basically what chromatography does is that we run samples, which are mixtures of a lot of different compounds through a column, and through their different diffusion properties, those compounds are going to diffuse through the column at different speeds, and so it's going to take them more or less time to go through the column, as you can see here at the bottom of those plots, what we call the retention time. And because there are different molecules, they're going to show up different times, and we're going to be able to separate them. And so, as you can see on those two plots, we found a lot of different amino acids. On the right plot, even in the same sample, Munoz Caro et al. In 2002 found 16 different amino acids in the same sample. So this is a liquid chromatography chromatogram that I had obtained in a residue several years ago. And as you can see, it's made of a lot of different amino acids. The simplest one is glycine and is the most abundant, as you can see. And then we have a lot of different others. And I'm going to show you this first. If you report the different amino acids that you find in these samples, you can see that the most abundant ones are the simplest one. Glycine is the simplest amino acids in proteins, alanine is the second simplest, and then serine, etc. And so this is also, to give you an idea, this is the total mass of those amino acids in one sample. It's measuring micrograms. And if you do the same experiments with different starting mixtures, different temperatures for different irradiation times, what you can see is that mostly the amino acids are made always in the same kind of manner with the same relative proportion, with glycine and alanine being the most abundant ones and serine after that. And the biggest amino acid being much less abundant. This is the plot I showed you earlier. And so if you draw a graph of the abundance of amino acid as a function of the molecular mass or the number of carbon, you can see that it decreases exponentially. And this is a proof that those compounds were made in a non-biological way, because in proteins for biological systems, this would be a little bit more balanced than this. And because here we have the simplest ones that are most abundant than the most complex ones. Something else that you can see by analyzing those residues for the search for amino acid with, for example, gas chromatography. In this case, we used a method that can separate enantiomers. As Dr. Aponte talked about like a few weeks ago in his talk, amino acids are chiral molecules, so they exist in two mirror image forms, or enantiomers. And biological systems only use the L form. But in this, for example, in this plot, you can see that we make L and D alanine, the two versions of alanine, with the same proportion, which is also an indication that those samples were not contaminated and that those molecules were formed in our experiments. You can use different kinds of starting mixtures, ice mixtures, to make those residues. For example, here, no methanol, just CO2, irradiate, and you obtain amino acids. You can even start with naphthalene, which is a small polycyclic aromatic hydrocarbon compound, and irradiate it in ices, and you will also make amino acids. So this process of making amino acid is pretty robust, no matter what the carbon source is. The second big family of molecules that was searched for in those residues is nucleobases. So nucleobases are the building blocks of DNA and RNA in all terrestrial life. And this is what where the genetic code is contained, actually, in DNA and RNA. And they are made of two basic structures, pyrimidine and purine. So I'm not going to go into details of this because Chris Marais is going to make a presentation next week about exactly that. And he's going to talk about all this experiment and the result in detail. The only thing you need to know that if you put pyrimidine in similar ices, you make all the compounds that are present in DNA and RNA. And if you do the same with purine, then you also make adenine and guanine that are found in terrestrial life. And I'm not going to talk about this anymore because Chris is going to talk about this in detail next week. The third family of molecules that we search for in residues are sugars and sugar derivatives. And so sugars play roles at different levels in life, from the building blocks also of DNA and RNA, but also as energy storage. And of course, you can find this sugar derivative in many other functions in biological processes. In meteorites, a lot of sugar derivatives have been found also. And until very recently, we thought that mostly only sugar alcohols and sugar acids were present. But I will show you that this has changed. And what is very interesting is that, of course, we find also those compounds in laboratory residues, as I'm going to show you. So until very recently, we thought that meteorites only mostly contain sugar alcohols, acids, and diacids, and only very small amount of sugars. But last fall, I think it was in October or November, this group from Japan, Furukawa et al., showed that at least two meteorites, Murchison and NWA 801, contain the sugar ribose and its three isomers, as well as the hydroxyacetone that was already shown by George Cooper in his publication. So meteorites do contain some of the sugars and sugar derivatives. So the search for sugar derivatives in residues hasn't started until very recently. And the reason for that is because this family of compounds actually consists of a lot of different compounds. And because their structure is very similar, it's very difficult to analyze and separate them. As you can see on this chart here, from only 3 carbon atoms to 6 carbon atoms, things get a little bit complicated. So the hydroxyacetone is the one that was originally found in meteorites here. And if you replace one group, the top group, the top aldehyde group with carboxylic acid, you get the same amount of sugar acids. And if you replace the top group here with like CH2OH, you get all the sugar alcohols. And what I want to mention here is that sugars are also chiral molecules, which means that here I show only the D enantiomers of all those molecules, but you also have all the mirror images of all those molecules also. So that's a lot of different compounds. And to make things even more complicated, that sugars like to be in different forms too. For example, they can be in a linear form, as I showed you before, but they can also be in cyclic forms, either in 5-member cycles or 6-member cycles if there's enough carbon atoms. And so that complicates the search for them even more. But we started to look for sugar and derivative in residues, and this is how we proceeded. We tried to develop a method to make sure that we can see what we were looking for, and it was not easy. But by trying different methods, we finally got to the point where we could separate those compounds pretty easily. And in this example here of a very simple mixture of water and methanol, we could find, for example, a lot of sugar alcohols from the glycerol, which has 3 carbon atoms, to up to the 3 sugar alcohols with 5 carbon atoms. If you zoom into that area here, you can even find other compounds such as sugars themselves with 3 and 4 carbon atoms, and also sugar acids with 3 and 4 carbon atoms. if you try to calculate how much of this compounds you have in this kind of residues, you will find that sugar alcohols are usually more abundant than sugars that are usually more abundant than sugar acids when you start only from methanol and water. And just to give you an idea, the total amount of glycerol in the samples is about what you can find in 1 gram of meteorites. And so if you start only from methanol in the presence of water, you can see that those compounds are formed just by adding CH2OH groups, and you can form a sugar alcohol, and by oxidizing this way, you can form the sugars, and then oxidizing even more, you can form the sugar acid. So basically the abundance of this compound decreases when you go from right to the bottom. Another group a few years ago found even ribose using even a more sophisticated GCMS method. And you can see here the peaks for ribose in its cyclic form. And as you can see here, they also found like the sugar alcohols and the sugars and the sugar acids in the same kind of decreasing abundance as I find in my samples. So that confirmed that sugar alcohols are supposed to be the most abundant ones. So something we haven't talked about in all those sugar derivatives that were found in meteorites on the right part of this chart here, deoxy sugar acids, which means, or deoxy sugar derivatives in general. They are mostly the same as the ones on the left part, except that they are lacking one OH group. And so for some reason, the protocols are a little bit different to look for them. And so we tried to look for those deoxy sugar compounds, and in several experiments, only starting again with water and methanol, both regular methanol and methanol marked with carbon-13, we actually found deoxyribose, which is the sugar of DNA. And we also found one of its isomers, deoxyxylose, but there's a question mark about this one because there's no commercial standard to compare our data to, but that's the only possibility when you have this configuration. This is the only other molecule that could be formed. So we think we could identify it. And very interestingly, in exactly the same sample, we both found deoxyribose and ribose, so both the sugars of DNA and RNA. In the same samples, we also found a bunch of deoxy sugar alcohol from 3 to 4 carbon atoms. And so I'm not going to go through all of them here. But what is very interesting is that in the same paper, we also analyzed 2 meteorites, 2 samples of meteorites from Murchison and GRA 06100. And we found a few of those deoxy sugar alcohols in those meteorites. Another family of molecules that is modern biology, at least, of long chains with a hydrophilic head and hydrophobic tail, and they are responsible for making membranes. And so we think that membranes are a very important step towards the emergence of life. And what is very interesting is that this kind of compounds were found in primitive meteorites and also in some organic residues from the lab too. So as you can see, there's like several kinds of those compounds that can form different kinds of vesicles or membranes. And the mystery is that we still don't know what those molecules are. And so one of the challenges would be to determine what compounds actually constitute those membranes. So and compare those found in meteorites and in the lab samples. And so yeah, this is an open question, and there's a lot of things to still discover in this field. The last thing we did with residues is that when we analyzed those residues with the X-ray absorption spectroscopy, is that when analyzing the samples using the X-ray beam of the synchrotron beam from the ALS, that we saw some chemistry happening in the samples while we were analyzing. And on this plot, you can see that the C4 band here, the band marked C4, which is aliphatic carbon, started to decrease when we exposed the sample to more X-rays, and at the same time, the C1 band, which is the aromatic band, started to grow. And so that was very interesting because we could see that alteration of those residues. And one thing we had in mind is that one part of the organic material that we find in meteorite is insoluble. So we call that the insoluble organic material, IOM. And what we think this material is made of is like a bunch of like aromatic material that is linked with aliphatic chains and contain also like other atoms such as oxygen or nitrogen. And so if we start irradiating those residues and start making more and more aromatic material, the question is, are we going to start forming something that looks like this or not? So to verify that, we started to make samples and so residues and to take them to the Synchrotron Radiation Center in Taiwan to irradiate them with high energy photons and see what we obtain. On the left right here, you can see samples with like a black mark. These are the samples that were irradiated, and the black mark is just where they got irradiated by the synchrotron beam. And so after we irradiate those samples, if you analyze them with infrared spectroscopy, so here on the left side is the whole broadband of X-rays from 80 to 450 EVs, you can see that the residue composition changes. And in particular, you have the growth of those two bands. And it's been like very interesting to try to understand what those infrared bands were, because they're not very common. And the closest I found in the literature is that those bands are attributed to nitrile groups that are linked to very highly aromatic material. So it looks like by irradiating those residues with X-rays, we are making more and more aromatic material, which is what we were looking for. On the right here is it's the same residue that was irradiated with one specific energy of the X-ray beam, and you can see that there are changes, but it's not as dramatic. Something else that we did with those residues that were irradiated with the synchrotron beam is to look for changes in deuterium content. See if those samples were enriched in deuterium after irradiation. And those are only like very preliminary materials. And so what we see is that for different composition of the residues and the fact that they contain naphthalene or not, you have a very wide range of enrichment in deuterium, but most of it looks like it's pretty significant. And very recently, we started a collaboration with another group in Taiwan to analyze the residues to search for composition in carbon-13, oxygen-18, and nitrogen-15. And it looks like we can see very small enrichments for those molecules as well. But this is very preliminary, so there's more to come later. And so to conclude the whole talk, if we can make all those molecules in those environments, astrophysical environments, basically we have all the ingredients we need to get life started on planets. So if those molecules are really made in those cold environments and they really rain down on planets when the planetary system formed, we have everything we need to start life. And so if this process exists everywhere, as we can see from observation, then this has important potential implication for the chances of rising in a lot of parts of the universe. And so to finish, I would like to thank all the people that contributed to all this work that I've presented, from people in France first, then in Taiwan, and currently here at NASA Ames, especially Scott Sanford and Chris Matarese, who moved to Goddard 2 years ago. And I would like to thank, of course, NASA for funding, especially Exobiology Emerging Worlds Program and the NASA Astrobiology Institute. And thank you for your attention, and we'll check if I have any questions. Oh, so I see the first question from Chris Bennett. Hi, Chris. From what I've checked recently, I haven't seen HMT detected in meteorites. There may be a reason for that. As you may know, usually to look for organics in meteorites, you need to process the meteorite samples with a lot of different steps, chemical steps. One of them is like going through acid hydrolysis. And as I showed in my slide earlier, that if you hydrolyze HMT, you make a bunch of other things, including amino acid. And so I'm wondering if HMT is actually present in those meteorites in some form or in any kind of variant of HMT, but then we just lose it when we hydrolyze the samples, and actually that contributes to the amount of amino acid that we find later after processing the meteorite material. So, second question from Carlos is HMT stable at room temperature? And if so, what is its absorption spectrum? So HMT is actually thought to form very close to room temperature. A lot of papers that I cited earlier show that actually HMT is not formed at low temperature in the ice. We only form the precursors to HMT, and they start to react together during the warm-up. And HMT actually forms closer to like 300 K at room temperature. And so, of course, it is very stable at room temperature. About the spectra, I would refer you to the paper I cited, or you can send me an email, and I will try to send you all the information I have about those papers. And so you can take a look if you're looking for the spectrum. Let me see. How do you interface your HUV chamber with a liquid chromatograph? So maybe I didn't explain that very well. The chamber is not connected to any chromatograph. What we do is that we warm up the samples from the chamber to room temperature, and then we take those samples out and actually analyze them with HPLC or GCMS outside of the chamber. So this is room temperature analysis. It's not inside of the chamber. The next question is from Rahul. Does this complex organic form during irradiation or during warm-up after irradiation? And the answer is probably both. Some of the compounds may form at very low temperature. The thing is that during the irradiation at low temperature, species are not very mobile. They are frozen. Basically, they don't move much, but they can still react with close neighbors. And so some of the molecules can be formed even at very low temperature. And I think there was a paper from the Kaiser group that showed that even like organic molecules with like 3 or 4 or 5 carbon atoms can actually form even at 15 Kelvin. But we suspect that a lot of those compounds are formed during the warm-up when the species start to move. And so they start to meet each other and start to react and form more and more complex molecules. Chris has a question about HMT. Haven't analyzed meteorites for a long time. I think that's a good point. And so if somebody has this technique and would like to search for HMT or its variants in meteorites, that would be a way to try. Thank you, Chris. So we have a bunch of questions from Mihai, if I pronounce it well. Do you have any mechanistic understanding of how this reaction in ice proceeds? For instance, you have an ice that contains water, methanol, ammonia, and HCN. As these reactions occurring in a neutral state, you produce a neutral radical that starts to react with the adjacent species, or do you have a mixture of radicals and ions? So I'm going to answer this question first. I think the answer is all of this. Because those compounds are frozen at really low temperature, I think the reason why they may interact is more easily with neighbors is because they are reactive. And for this is because they are either ionized or some radicals are formed. And in this case, you don't have any energetic barrier for reactions. And so those can happen more efficiently, even at very low temperature. And so we think that, yeah, that all those different reactions actually occur, like radicals and ions, and radicals with ions, and radicals with radicals, and radicals with neutrals. So I would say a little bit of everything. What is the role of the substrate? Does it play any role on the whole reaction? So in our experiments, I don't think the substrate plays a big role, because maybe only the first layer, the first layer that is in contact with the substrate may have different properties. But after that, we just accumulate like sample all over the substrate. So most of the ices or the photo products formed from the ices do not see the substrate at all. So I don't think it really matters in our experiments. Did you try experiments at different coverages? So if you mean that if we try different substrates, the answer is yes, and it doesn't really change anything. Again, using the protocol that we use for our experiments, it doesn't change anything. Third question was, do you expect a completely different reaction if you irradiate the ice sample with UV, XUV, or electrons? So, so some other groups have irradiated very similar ices with different energetic sources. For example, even higher energy UV photons, or energetic electrons, or even energetic protons. So the processes in the ices may be a little bit different, because the way the energy is distributed inside of the ices is different. For example, UVs cannot penetrate that much deeper inside of the ice, but for example, energetic electrons can go deeper, or energetic protons can go deeper. But what is very interesting is that in one of the papers from Jerikens, I think in 2001, if I remember, where they irradiated exactly the same ice samples with UV and energetic protons, even if the chemical process itself is different, the residues at the end look pretty much the same. And so basically it means that as long as the ice is frozen and you bring energy to the system, chemistry will happen. And in the end, if you just have compounds with carbon, oxygen, hydrogen, and nitrogen, you will form the same kind of compounds in the end, no matter what the energy source is. I'm less familiar with X-rays. I know some groups have recently done experiments also in Taiwan about irradiating simple ices with X-rays, but I haven't looked into those like in detail. So I will take a look. Thank you. What is the UV photon energy and the flux? So mostly the lamp that we use emits Lyman alpha photons, which is about 10.2 eV, and a continuum a little bit at lower energy, so down to about 7 or 8 eV. And the flux is about, it's estimated about to 10 to the 15 photons per second per square centimeter. Next questions. If all these essential life-building materials can be synthesized abiotically, how can we distinguish them from a real-life signature in space? Well, that's a very, very good question. But since we don't know how life elsewhere will look like, it's even more difficult to answer that question. We can make the distinction pretty easily between biological materials from Earth and what we do in the lab, because as I showed, if you make amino acids in the lab, mostly you're going to make the same amount of both enantiomers of each amino acids. But biological systems, we amino acids, so the proportions would be very different, and this is how actually we try to estimate how much contamination the sample has, and so that's one way. But then in space, if it's a different kind of life built with different building blocks or something similar but with different proportion, we have no idea what it's supposed to look like. So we'll have to make our best guess based on very detailed analysis, but it's very difficult now to answer that question for sure. So Katerina has questions. How does the UV flux of the lamp we use compare to the UV flux received in the body? I think I put that in one of the slides earlier. In terms of dose, 2 days of experiment is about 10 million years of exposure in dense molecular clouds. And of course, this depends on the density of the medium. If it's a more diffuse medium, then it will be like a shorter time because photons can penetrate more easily. Do you think the timescale formation of these organic molecules in the lab compared to timescale or on interstellar ice bodies on Pluto? So on Pluto it would be very different though, because the UV flux is different, and I don't have the numbers right now, but I'm wondering if Chris Matteries in the papers I cited earlier from 2014 and 15, there may be an estimate of how much time of irradiation this corresponds to compare with the lab. And the last question was, have you tried irradiating ice with polarized UV light to see if you can create an enantiomeric excess? And the answer is yes. I tried that a long time ago and it was not very conclusive, but a group has done a better job after me a few years ago, and they could create enantiomeric excesses in alanine and maybe another amino acid using circularly polarized light. So I can send you the links to the papers, or send you the papers if you want. But yes, the thing is that to achieve that, you need to destroy a lot of material. And so the more excess you are aiming for, the more material you need to start with. Otherwise, you don't have anything left. So, and of course, it becomes more and more difficult to measure if you have less material. So it's kind of tricky. But yes, such experiments were done before. So next question from Sergio. As you showed in your simulated experiments, and there were amino acid mixtures of L and D forms. Most terrestrial amino acids are L form. What are your thoughts about this terrestrial phenomenon? Do you expect life in other planets' icy to follow the same rule? I wish I could answer that question. I think that's something everybody wonders, why terrestrial life is made of mostly L amino acids. I think from a biological point of view, it makes total sense to have only one form. It's actually one of the keys to biochemistry. But the reason why we have only L and no D is an open question. Like a lot of different groups have analyzed meteorites, and when you analyze the meteorites to look for amino acids in those meteorites, some of the amino acids show like excesses of L. It's pretty small. It's not 100%. It's like it goes up, I think, to 15 or 20% for some amino acids, but it is there. And so it looks like maybe this very small excess of amino acid in meteorites could have influenced the fact that life only chose amino acids of the L form because they were just more abundant. But there's no real answer to this question yet. It's still like a topic in debate right now. And as of other life on other planets, I mean, it's an open question too. Since we have only one example of life, and it's life as we know on Earth, then it's very difficult to extrapolate. But if chirality is a necessity, like a requirement for biochemical reactions to happen, then I guess maybe it would be similar, but that doesn't mean that it would be based on L amino acids. Maybe in different, like a different stellar system for exoplanets, maybe the D form is more abundant, and so life started on D amino acids. So it's very difficult to answer that question. All right, I have a question from Patrick. Would you expect to see some of the sulfur-containing amino acid in your organic residues if you included thioderivative in the initial ice composition? So to tell you the truth, we tried that several years ago, and it's not easy. And I looked for those sulfur-containing amino acids, and I couldn't find them. That doesn't mean they're not there, but it looks like it's a little bit more complicated to transfer like sulfur atoms from, if you start, for example, from H2S or SO2 in the starting mixtures. It looks a little bit more complicated to transfer that. Or maybe we make like compounds that look like amino acids and that include also sulfur atoms, but not the ones that are not the same compounds as the amino acids that we know of. And so I didn't look for other compounds. I only looked for the two, cysteine and methionine, I think, and we couldn't find them. But that doesn't mean they're not there, and we haven't repeated the experiments for a long time. So maybe it would be worth looking for them. Ricardo, hi Ricardo, has a question. So it's in residues. Do you observe relevant differences according to the irradiation dose given to pristine ice material? So the dose, sometimes I could see differences and sometimes I couldn't. It depends how much you vary the dose. Basically, if the dose is too low, then reactions happen, but then you end up with a residue that is smaller and doesn't contain as many, for example, amino acid or sugar derivatives. But if you irradiate with a higher dose, you may start to destroy what you make. And so there's probably a pretty wide range of doses from which you can make all those amino acids and sugar derivatives. But at some point, if it's too high, you may start to destroy them. And I haven't explored this parameter as much, so I can't tell you how much is too much. But that's a very good question, and that would be a very interesting thing to study in the future. Thank you. Question from Brian. Have you attempted any warming cooling cycles during irradiation to mimic orbit? If yes, what effect does it have on the residues form? Some kind of such experiments have been performed, and I think I did a couple of those experiments a few years ago. And basically what happens is that if you do your experiments at low temperature and you warm up, you're going to get rid of all the volatile compounds, and you're going to end up with the residues. But then as you cool down again, then it's going just to be covered with more ice, and the whole thing is going to start all over again. So the only difference would be that under the ice, there would be like a refractory material made by the residue. And some, I think I read that somewhere, some people show that sometimes this residue can behave as a catalyst for the few layers of ice that are in contact with it. And so it may boost the formation of organics a little bit, but not that much, because only the layer of ice that is close to this residue will be enhanced. And so so I think more work should be done about that. But mostly people would do that maybe to try to optimize or to increase the amount of organic material that you get in the end, which is easier to analyze. Also, you can find more smaller species if you have more material. But yes, that would be another parameter to explore more in detail. I think I've been through all the questions. So
all right.
Anybody has any more question, I think, yep.
So I think I'll pull up the concluding slides then.
Mm-hmm.
Okay, so just once again, thank you so much to Dr. Nuevo for the excellent talk. And as always, our audience for joining us and asking so many thoughtful and interesting questions. So coming up next week, we are continuing the ice irradiation talks with Dr. Andrew Mattiota on Tuesday, presenting on the wonderful universe of PAHs. And on Thursday, we will hear a talk from Dr. Christopher Matarese on the formation and destruction of nucleobases and ices, as Dr. Nuevo mentioned. So if you like Dr. Nuevo's talk, please join us again next week. And we'll see you guys later.